Once a neurotransmitter is synthesized and packaged into vesicles, it is ready to be released. And so release is triggered by an electrical signal, usually, and that electrical signal is usually the action potential. So we talked all about the action potential in the last chapter. So usually the action potential starts at the cell body or at what's called the spike initiation zone, and it travels down the axon to all of the axon terminals. So if an axon has multiple collaterals, each of those collaterals is going to eventually end in a terminal, and that action potential will travel to all of them. So when you get down to the axon terminal, you still see these. Here are voltage gated sodium channels in the membrane. And so as the action potential travels down, it opens more and more of those channels. And when they open, they let sodium go in. So here's sodium coming in. And so we already know what that means. That means that the membrane depolarizes. So when the action potential gets to the terminal, it makes the inside of the cell, the inside of the synaptic terminal, more positive than the outside. And that triggers the opening of a type of channel called voltage-gated calcium channels. So calcium is another ion, and it has to get into the cell through a channel just like sodium. But calcium, and calcium like sodium, also is more concentrated on the outside than the inside. In fact, there's a much greater concentration gradient for calcium than there is for sodium, which means that the ionic driving force for calcium is much larger. Usually the equilibrium potential for calcium is something like 120 millivolts. So when you open voltage-gated calcium channels, they will, of course, come in, just like sodium does. And when calcium comes in, that actually is the trigger that causes the synaptic vesicles to release their neurotransmitter. And so that is a kind of complicated process. We're not going to go into too much detail, but just to explain, calcium ions, or if you look at the vesicles, they are anchored to the membrane by this complex of several different proteins. So there's a protein that's embedded in the membrane called T-snare. T stands for terminal, and then that binds to a protein that's embedded in the vesicle membrane called V-snare. So that's what actually locks the synaptic vesicle down to the presynaptic terminal membrane. And so when the neurons are at rest, or there's not a change in membrane potential, the vesicle just sits there next to the membrane. But then there's another protein called synaptotagmin that's part of this complex, and it is sensitive to calcium. So when calcium concentrations go up, the synaptotagmin protein binds to calcium, and that triggers a reaction between V-snare and T-snare, causing them to both shift around such that the vesicle fuses with the membrane. So now the membrane that was the vesicle, so here's the membrane vesicle, and here's the presynaptic terminal. Here's the membrane of the vesicle and the membrane of the presynaptic terminal. And then synaptotagmin activation triggers those two to fuse. So now the membrane of the vesicle and the membrane of the terminal become one, and that means that the contents of the vesicle become free to leave and become part of the extracellular fluid, and that process is called exocytosis. So exocytosis is a generic term for any time something inside a cell is released like this, like with a vesicle that fuses with the membrane and releases its contents. So this isn't actually just for neurotransmitters. There are other substances like hormones, for example, that are released from glandular cells in a pretty similar process. Then, once the neurotransmitters are free, or once they're released from the vesicle, they then have the ability to diffuse across the synaptic cleft. So remember this gap we call the synaptic cleft, and it's very short, so it's only a few nanometers across. So the molecules don't have to go very far, but they diffuse across into the extracellular fluid, and then eventually they bind to their receptors. So these are the receptors down here. We'll talk about them more in a second. and then the vesicle itself actually then reforms. So once the neurotransmitters are released, this chunk of membrane now that includes the what used to be the synaptic vesicle essentially pinches back off into the cell and forms a new, now empty vesicle. And that process is called endocytosis. So this is how vesicles kind of get recycled. So eventually, then they can be refilled and can then go back to the reserve pool, and then eventually go through the whole cycle again. And again, this happens in other cells too. So anytime a cell takes up any sort of molecule from the extracellular fluid, it can be done through a process similar to this also. So now let's talk about the neurotransmitter receptors. So these are the proteins that are found on the postsynaptic cell that are going to respond to the neurotransmitter. So again, this is the presynaptic side in blue, and then down here is the postsynaptic membrane. And again, this can be part of a dendrite, it can be the cell body, it could be anywhere on the neuron, or you could even not be a neuron. You could be talking about a muscle or a glandular cell. So when we talk about the postsynaptic membrane, that's what we mean, just the membrane of whatever the cell is that the presynaptic terminal is forming a synapse with. And the neurotransmitter receptors, these are the receptors, these orange things, are proteins, and they are embedded in the membrane of the postsynaptic cell, and they have specific binding sites for the neurotransmitters. So again, these are the neurotransmitters. And so when they're released, they can travel across, and each of these receptor proteins has a location on somewhere on its surface that fits that receptor perfectly. In other words, it's almost like a lock and a key. So the neurotransmitter is like the key, and the receptor is like the lock. There's a surface, a position somewhere on that protein that it precisely has a place where the neurotransmitter can stick to, and only that neurotransmitter. So in other words, if a different molecule comes along, it won't activate these receptors. These receptors are specific for the neurotransmitter being released from the presynaptic cell. So if this is for example, if this is a glutamate synapse, so that means that glutamate is the neurotransmitter, glutamate's being released from the presynaptic cell, then the receptors on the other side have to be glutamate receptors. There would be, you wouldn't see, dopamine receptors, for example, or serotonin receptors at a glutamate synapse. They would only be glutamate receptors. Again, you could have also peptide neurotransmitters, and so there would also be receptors for those peptides as well on the postsynaptic side. But at the very least, whatever's being released from the presynaptic cell, there has to be specific receptors on the postsynaptic side. And the receptors come in lots of varieties. There are, for every neurotransmitter, in fact, there can be sometimes many, in some cases, hundreds of different types of receptors. And they can be classified different ways. They cause the cell to respond in different ways. But broadly, we can classify them into two groups: the ionotropic receptors and the metabotropic receptors. Ionotropic receptors, as we're about to see, are basically neurotransmitter-gated ion channels, whereas the metabotropic receptors are proteins that do not act as ion channels, but they still trigger an intracellular response by activating a cascade of other enzymes and proteins. So we'll talk about the ion channel, the ionotropic receptors first. So again, these are all neurotransmitter-gated ion channels, and that means that they are gated by the neurotransmitter. So they only open when the neurotransmitter binds. So again, if the neurotransmitter is here, if these are our neurotransmitters in blue being released, they will bind to the receptor. These are the receptors, the brown ones, and when they bind to those proteins, they cause them to change their conformation such that they open a channel that lets ions go through. Now, just like the voltage-gated ion channels that we talked about in the last chapter, these are closed until the neurotransmitter shows up. Unlike voltage-gated channels, though, generally the neurotransmitter-gated ion channels are sometimes less picky. So, for example, we call this a neurotransmitter-gated sodium channel, because here we're showing sodium going through, but you can have neurotransmitter gated channels that will let other ions go through as well. We'll talk about that more later. But generally, ion channel receptors, when they open, they let ions go either in or out, depending on the type of ion. And they then change the membrane potential, because of course that's what ions do when they go across the membrane. They either make the membrane potential go up or down. And of course, you should know in this case what would happen if, let's say, we let sodium in. So if this neurotransmitter is activating a sodium channel, then you should be able to guess what will happen, because again, in a typical cell, the sodium concentration is much lower on the inside than the outside, which means that for most neurons, they have a large positive equilibrium potential for sodium. And so if you open a sodium channel, sodium will come in, and that will raise the membrane potential, or what we say is that it will depolarize the membrane. So if you have a neurotransmitter gated sodium channel, most of the time it will depolarize the membrane, and that's what's happening here. So here's a graph of membrane potential over time in response to neurotransmitter release, or in response to the neurotransmitter gated channels opening. So when the channels open, the membrane potential becomes more positive. So and how high it goes depends on how many channels are open and for how long. Unlike voltage gated sodium channels, which have a very predictable opening, closing, and opening and inactivating pattern, these channels will stay open as long as there is a neurotransmitter bound to them. So that means that their profile, in other words, their effect on the cell, can be sort of is smaller. In other words, the height of this peak may be shorter, but it's sort of dragged out because these channels will stay open as long as the neurotransmitter is present. And any change in the membrane potential of the postsynaptic cell we call a postsynaptic potential. So a postsynaptic potential is what it sounds like. It's just a change in the membrane potential of the postsynaptic cell. And usually when you have a sodium channel opening and you have the membrane becoming more positive in response, we call that an excitatory postsynaptic potential, because this, and we'll learn why this is later on, but this membrane potential, this change in membrane potential, would have the ability to trigger more action potential. So if this cell, if the postsynaptic cell is a neuron or a muscle cell, then this change in membrane potential would have the potential to eventually trigger an action potential in the postsynaptic cell. So that's why we call it an excitatory postsynaptic potential, or oftentimes we just abbreviate it EPSP. So when you hear EPSP, that's what it means: excitatory postsynaptic potential. A depolarization in the postsynaptic cell caused by the opening of neurotransmitter gated sodium channels. So some neurotransmitters have these kinds of channels associated with them. Glutamate is a good example. Most of the glutamate gated ion channels are sodium channels, or calcium, and those all cause depolarization. So typically glutamate we call an excitatory neurotransmitter, because it usually opens excitatory or neurotransmitter gated sodium channels. Meanwhile, if you have a neurotransmitter gated ion channel that's permeable to chloride, then when the neurotransmitter is present and it binds to the receptor, it will allow chloride to cross over. Chloride, remember, typically has a negative membrane potential, but the concentration is the same as sodium. So that means that the concentration for chloride is usually greater outside the cell. And so depending on where the membrane potential is, if the membrane potential is less negative than the equilibrium potential for chloride, then chloride will come in. If it's more negative, then it will go out. But the point is, just like any other ion, whenever you increase the permeability of the membrane to that ion, you're going to drive the membrane potential closer to the equilibrium potential to that ion. So opening a bunch of chloride channels will force the membrane to stay closer to the equilibrium potential of chloride. And typically, that's either going to bring the membrane potential down. So if chloride ions are entering the cell, that means the inside of the cell will become more negative, which means it's going to hyperpolarize. Or if the membrane is already at the equilibrium potential for chloride, then the membrane potential won't change, but it also will be harder to increase, which means that if you have a depolarizing current, let's say from a different synapse where you have sodium coming in, then it will take more depolarization to trigger an action potential. Either way, you're reducing the likelihood of an action potential because you're either lowering the membrane potential or you're keeping it close to the resting potential. But in either case, we call that inhibitory because it's preventing the cell from or making it harder for the cell to produce action potentials. Not impossible. So you can have, if the amount of excitatory postsynaptic potential is greater than the inhibitory postsynaptic potential, you could still have action potentials. But that's what the inhibitory postsynaptic potential means, or IPSP. It's just either a hyperpolarization of the membrane in the postsynaptic cell, or just a counter hyperpolarization to a depolarizing current from a different synapse. But in any case, if this is the effect of that neurotransmitter, then we say that this neurotransmitter would be inhibitory. So for example, GABA, that's one of the other amino acid neurotransmitters, and glycine also, typically open, if they open transmitter-gated ion channels, they are chloride channels. Most of the ionotropic receptors for glutamate and I mean for GABA and for glycine are chloride channels. So those two are usually inhibitory receptors. But again, it's not the nature of the neurotransmitter, it's the nature of the channel. So the only reason we say that GABA is inhibitory is because it typically causes the opening of GABA-gated chloride channels. So it's really because of the channel or the receptor that we say that it's inhibitory, not because of the neurotransmitter. If you had, and you could, there's no reason why you couldn't have a GABA-gated sodium channel, then in that synapse, GABA would actually be excitatory. Now, the other major type of neurotransmitter receptors are called metabotropic receptors, or sometimes they're called G protein coupled receptors, or GPCR. And the reason they're called that is because they are on the inside of the cell, they're attached to a different protein called a G protein. Okay, now this is important. A lot of people get confused about this part. The receptor is called a G protein coupled receptor. That is the protein that is embedded in the membrane that actually attaches to the neurotransmitter. Again, here's the neurotransmitter, and here it is binding to the receptor. The G protein is a separate protein that's on the inside of the cell. It does not cross the membrane, and it does not interact with the neurotransmitter in any way. It can only be activated by the receptor. So the way the metabotropic receptors or G protein coupled receptors work is the neurotransmitter binds to the receptor, and then the receptor activates the G protein. And then, and importantly, the receptor is not an ion channel, so no ions need to cross the membrane or cross the membrane through this protein. It's just stuck in the membrane, and it's only affected by activating the G proteins. And then the G proteins then go on and activate other things. And there's a whole bunch of different pathways that G proteins can interact with. So for example, there are G protein gated ion channels. These are ion channels that are embedded in the membrane that, again, do not bind to or interact in any way with the neurotransmitter, but because they're activated by the G protein, it's a different pathway for the neurotransmitter to change the membrane potential. Again, these can be either excitatory or inhibitory, depending on the type of channel, but it's an indirect connection between the neurotransmitter and the membrane potential. Other G proteins, and even sometimes the same G protein in the same cell, can simultaneously activate another pathway called a second messenger system. And so a second messenger is a different molecule that's made inside the cell. So the neurotransmitter itself is the first messenger. The second messenger is something that's made inside the cell by an enzyme, which is usually attached to the membrane, that's activated by the G protein. So the G protein activates the membrane-bound enzyme, and then the enzyme makes the second messenger molecule. And there's a whole bunch of different second messenger systems. Each one then activates its own downstream pathway. So there's a whole complicated network of molecular signals that gets triggered by these second messenger systems. So the second messenger, or the metabotropic receptors, are way more complicated than the ion channel receptors. So the ion channels, they can only change the membrane potential. That's it. They can either make the membrane potential go up or down, or stay where it is. And that has an immediate short-term effect on the cell. It either triggers an action potential or prevents an action potential, depending on the change. Whereas metabotropic receptors can have a whole bunch of complicated downstream effects depending on which pathways are activated, and sometimes it can cause changes in the membrane potential. Sometimes it can cause other more complicated things to happen. So you can have more receptors added to the membrane or taken away. Some second messenger systems will trigger gene expression pathways, so it can actually cause the cell to turn on or off certain genes, completely changing its behavior. So these metabotropic receptors, we'll talk more about them later in a little more detail, but rest assured they're way more complicated. Now, we're not quite done with the transmission process, because the neurotransmitter was released from the presynaptic cell. It diffused across the cell the synaptic cleft to the receptors on the postsynaptic side. But now the neurotransmitters need to leave, because the neurotransmitters will, again, stay there, and as long as they're there, they will keep activating those receptors. They will keep the ion channels open. They will keep the metabotropic receptors active, and that's not a useful information transmission system. In order for something to be useful for transmitting information, there has to be a way to turn it off. So most neurotransmitters have some sort of removal method, some way of getting the neurotransmitter out of the synaptic cleft so that it no longer activates the receptors. Typically, so when a neurotransmitter binds to a receptor, even though there's a relatively high affinity between the receptor and the neurotransmitter, eventually they just let go. So just due to the way that molecules interact, eventually the receptor and the neurotransmitter part ways. And again, if the neurotransmitter is not removed, it will just diffuse and perhaps bind to another to another receptor. So we have to get rid of it. So there's two different ways to remove neurotransmitters from the synaptic cleft, or two different ways that are common. So there are specific enzymes for some neurotransmitters that break the neurotransmitter down into its component parts. Actually, the only neurotransmitter that this really applies to is acetylcholine. So, and we'll talk about this more later, but acetylcholine is removed from the synaptic cleft by an enzyme called acetylcholine esterase. And so that's what this enzyme is called here. This is acetylcholine esterase, which is actually abbreviated ACE. And so acetylcholine esterase breaks acetylcholine down into choline and acetate. So, and what's important is that those two molecules separately do not bind to the acetylcholine receptor. So it has the effect of removing the neurotransmitter from the synaptic cleft. For pretty much all other neurotransmitters, though, the neurotransmitter gets taken back up into the presynaptic terminal in a process called reuptake. So reuptake just means sucking the neurotransmitter out of the synaptic cleft and bringing it back into the cytosol where it came from. And this is usually carried out by specific proteins called reuptake transporters. These are proteins that just move the neurotransmitter out of the synaptic cleft back into the cytosol. And they act like the vesicular transporters we talked about earlier, which are the proteins that package neurotransmitter into the vesicles, but in a way they actually work backwards. So whereas the vesicular transporters move neurotransmitter out of the cytosol into the synaptic vesicle, the reuptake transporters move them out of the extracellular fluid into the cytosol. Also, there are some of these neurotransmitter transporter proteins on astrocytes. So one of the jobs of astrocytes, as we talked about, is to kind of regulate the extracellular fluid. And so one of the ways they do that is excess neurotransmitter can be removed from the extracellular fluid by the astrocytes. So they have their own transporter proteins that can remove those molecules. Now, one more thing to say about neurotransmitters is that they are just molecules, and they are molecules that are made by the neurons that release them. And they bind to their receptor just like any other kind of receptor ligand combo. And that's how all proteins work generally, is by binding to other molecules. And so most proteins have specific molecules they're supposed to bind to called ligands. And so the neurotransmitter in the case of this system is the ligand. But anytime a protein binds to something else, especially if it's a receptor, we call that something else the ligand. And the ligand is something made in the body, in the cell that it's released from usually. And so we call it an endogenous ligand. Endogenous just means it comes from the inside. It's generated in the cell or in the body. And so that's the thing that's supposed to bind to the receptor. However, it is the case that most receptors and most proteins can bind to other molecules that are not from inside the body, and those are called exogenous molecules. And in fact, most drugs and poisons and things that come from outside of your body that have some effect on your body are molecules that bind to receptors or other proteins and change their behavior in some way. And that's true of neurotransmitter receptors also. Generally, when something from outside the body comes in and binds to a receptor, it either stops the receptor from doing its job, either it blocks the neurotransmitter, or it somehow in some other way prevents the receptor from working. And if it does that, we call it an antagonist. So an antagonist is, again, some molecule that comes in and stops in this case, a neurotransmitter from binding to its receptor or prevents the receptor from functioning. But you can also have exogenous molecules that come in and bind to a receptor and actually mimic the effect of the endogenous ligand or the neurotransmitter, in which case we call that kind of molecule an agonist. So again, an antagonist prevents a protein or a receptor from doing its job. An agonist is something that mimics the effect of the endogenous ligand, and in fact, in some cases, may even be more effective than the endogenous ligand. So again, a lot of drugs, medications, and poisons, environmental toxins, these are usually chemicals that bind to these receptors in one way or another and cause the endogenous receptor to behave differently. And this, again, is not just true for receptors. The same thing goes for the transporter proteins. So vesicular transporters and reuptake transporters, they also can have different antagonists and agonists. And in fact, sometimes we name these proteins after not the endogenous ligand, but by the exogenous ligands that have experimentally been discovered to bind to them the most effectively. And we'll talk about those more in the next chapter. So in the next video, we'll just talk about some of the things that happen in the postsynaptic cell after the receptors have been activated.